everybody to the second day of the International School of Bioinformatics and Computational Neuroscience 2016. We're extremely delighted to enjoy your presence here today. The first conference is in charge of the Professor Giorgio Ascoli from the George Mason University, United States. He holds a bachelor's degree in chemistry from the Scuola Normale Superiore of Pisa, and also holds a PhD in biochemistry and neuroscience from the Scuola Normale Superiore of Pisa. He's the leader of the Computational Neuroanatomy group in the Center of Neuroinformatics, Structures and Plasticity at the Krasnow Institute. Dr. Ascoli is also the founding director of the Center for Neural Informatics, Structures and Plasticity. His work is centered in the connection between the cellular organization of the brain and the cognitive functions that such networks display. He also is interested in how some network features of the brain, such as topology and its connectivity, are related with its electrophysiological activity. Professor Ascoli has also lectured extensively at the George Mason University. So please, let's welcome the Professor Ascoli with a generous applause.
Can you hear me fine? It's a great pleasure to be here. My first time in Bogotá, my first time in Colombia, for that matter. I found that understanding a little bit of Spanish is a lifesaver. So if you want to ask questions, you can try in Spanish as well as in English. But I'm going to try to keep the next 55 minutes or so very interactive. I have a completely hands-on approach to neuroscience. I like data. I like to play with data. I think that the traditional way of doing research, which is to have an idea, to create a hypothesis, to go to the lab, test it, and so forth, is not the only way to do science. I think there is a lot of playfulness in science, just playing with the data and get inspiration from the data. And I think that bioinformatics has taught a lot to us in this regard. I think the more complex the system is, the more playing with data becomes important. And the tools that I want to illustrate for you today are tools for investigation and exploration of basic data in neuroscience, specifically the structure of neurons and the organization of circuits using the hippocampus as an example. So please stop me anytime. I have no problem. It's not a sequence of logical steps. I'm going to jump from many nodes in the conceptual network that we're going to explore. So we can start at the end and end at the beginning. It doesn't matter. You can ask any question at any time, and I probably can get to it immediately. So please don't be shy. So we're interested in the brain and understanding how the... Yes? Oh, you totally can. Yes, yes. And in fact, if you want, in the end, I only have a few slides. The rest is all going to be websites. Feel free to pull out your own device, smartphones, computers. You can try to connect yourself. The Wi-Fi is okay, so you should be able to follow even live on the stuff that I show. But yeah, the slides are, I have maybe 10 slides. Feel free to take pictures, or if you want to email me at the end, I'm happy to send you a copy of the slides. So the brain is what we're interested in, and like all other biological objects, is made out of cells. But here is where things get interesting, both in terms of numbers and, in fact, the specificity of the features of these cells. So if you look at a slice of a brain, we put it under a microscope, and we start magnifying it. If we stain it properly to see cells, we can see a bunch of cell bodies. And those look a lot like cells in many other organisms, like the liver or the kidney or the skin or the blood, and so forth. Not only that, but these panels are in fact real data from available atlases online, and they are taken from different parts of that monkey brain. And you can see that even if I took slides from different places in the brain, all the cells look the same. However, if we are able to now look at the fine structure of these cells, we'll see that the story becomes much more interesting. because the cell body is only about 1% of nerve cells. So when you actually measure the surface area, the amount of membrane, the cell body of a kidney cell is all that that kidney cell has. The cell body of a liver cell is all that that liver cell has. But the cell body of a neuron is only about 1% of what that neuron is. And if you're able to look at the neuron in proper ways, which we now have great techniques to do so, you'll see that the neuron is made out of trees. And in fact, we now know there are two kinds of trees, and I'll speak plenty about those two kinds, which are basically working as inputs and outputs. And this is in fact where most of the complexity of the brain lies, because if you now look around in different parts of the brain, even within one part of the brain, you'll see that the trees are very diverse from each other. So you can think of botany as a great metaphor, okay? Real tree. So there is a park just next to this beautiful campus. You can walk in the park, which I plan to do this evening once the conference ends at 4. I'm going to take a walk, and if you come and take a walk with me, you'll see that, of course, no two trees are exactly alike, right? You can look at, say, 5 pine trees, and no one will be exactly like the next one. However, if you keep walking in the park, you'll see there are also trees of different types. So you might see pine trees and oak trees and cedar trees and palm trees, etc. And now also, the trees that you find here in Bogotá might be different from the trees that you'll find in Washington, D.C., where I live, or in Milan, Italy, where I come from, right? And so it's the same in the brain. In a given region, you'll find many pyramidal cells, many stellate cells, many basket cells. The pyramidal cells are going to look more similar to each other than to the basket cells and to the stellate cells. But then the cells in the visual cortex are going to be different from the cells in the, say, spinal cord. Okay? And this is just an illustration of many of the shapes of trees that you find in the brain. And I'll show you, in fact, the website, neuromorpho.org, where you can literally download, explore, quantify, model, simulate, and analyze, visualize, play with tens of thousands of these neuronal reconstructions. Please stop me anytime, okay? So those are the cellular constituents. And now let's look at what a human brain has. So the key thing is that the neurons use these trees to make connections. So the brain is a network. Okay, that's the most fundamental thing about the brain. It's a network. And the connections between neurons are called synapses. And those are made typically from the axon, so a tree, an output tree of one neuron called an axon, and the input tree of another neuron called the dendrite. Okay? So when two neurons connect, the axon of the sending neuron connects to the dendrite of the receiving neuron. Okay? And here is where things get even more interesting, because in a human brain there are of the order of 100 billion neurons and approximately 10,000 connections per neurons, which means 1,000 trillion synapses. And that's a very large number. So that's about the same order of magnitude than the number of real trees in the world, right? So the number of neurons in a human brain, if you were to walk around, not just the park next to this campus, but all the parks in all the planet Earth, you'll get about the same number of trees. And the number of synapses is about the number of leaves that you could count on the planet at any one time. Now, when you look at the structure of this, you discover many interesting things, which is that the branches of these trees are extremely small. Now, when you think biology, extremely small shouldn't surprise you. But the interesting thing is that the trees themselves are not small at all. You have trees of individual neurons that travel from the top of your head to the bottom of your back. So the length of those trees is literally macroscopic. However, they are as thin as 1/1000th of one of your hairs. Which means that even if these trees can be very long and very complex and very beautiful, I'll show you plenty of examples of those, you can pack many of them in the brain. Okay? And the total number of a single neuron tree, so we're not using scaling here, we're just doing that, is then can be measured in centimeters all the way to maybe decimeter. I think the longest can go into meters. Okay, so I'm using the imperial system here because I come from the United States. I don't know if you here use the international system, so meters instead of feet. And so when you actually count, you say, okay, what is the total length of the cable, right? If I were to unwrap all the cable, right? Now, you can read that if you unwrap all the cables of an airplane, like the one that I took yesterday to fly here, you get, I don't know, like 48,000 miles of cable, and you imagine all this very complex wiring of electricity, right? But in a single human brain, you get something like 150 million miles, so about more than 200 million kilometers. That's a lot of mileage, okay, in a single brain. So that means that if you take Google Maps and you look at all the roads that Google Maps has, all the paved roads in the world that are visible from a satellite, that's about the length of the cabling in a single human brain, okay? So you can go around the equator 5,000 times to cover that length. And so I was on the plane yesterday figuring out at what speed we were flying, and it would take 15 years flying at that speed with a pencil attached to a piece of paper to trace the length that long. Okay? That's in a single human brain. So these trees really are mighty. They are big, and they use their length and their complexity to connect with each other. And ultimately, I believe that the structure of that connectivity, the topology of that network, can explain how the brain works. So I'm going to spend a few more minutes just covering the basics of dendrites and axons so that you can get the foundations. And then we'll jump into the live demo with the data. So again, if I'm saying things that you know already, just bear with me in a few minutes. I just don't want to lose anyone. So axons are the output trees. So every neuron typically has dendrites and axons. So the axons carry the output from the presynaptic neuron to the target. And the dendrites receive and integrate the inputs from other neurons. Now remember, a neuron typically connects to 10,000 other neurons. So typically the axon of a single neuron will connect to the dendrites of 10,000 other neurons. And the dendrite of a single neuron will receive connections from the axons of 10,000 other neurons, on average. Some more, some less. So the dendrite is the input. Now the axon covers most of the distance. So check it out. If you look at this dendrite here and this axon here, on scale, this is the same dendrite. I just put the scale in so that this is exactly the same image in scale. So the dendrites are already much bigger than the cell body. You see the cell body is green here. That's what we could see in the previous picture when it was purple. So the dendrites are already much more extensive than the cell body. But the axons are huge. In fact, you don't even see the cell body here, okay, compared to the dendrites. Do that make sense here? So axons carry the signal. They actually do the job of communicating. The communication is the axons. Okay? However, the dendrites are the smarties. They are the ones computing. Okay? So the axons just sends the signal. Doesn't do anything to it. 0, 1. But it sends it long distance. The dendrites takes these tens of thousands of zeros and ones and runs very complex computations. Not unlike the complex CPU of a modern computer. But that's one single neuron. Now think of a massively parallel computer that has 100 billion of them. Okay? So the axon uses, to a first approximation, a binary code. Okay, so they go as they are throughout the entire tree, whereas the dendrites manipulates the signal. It can use very small or very large electric signals to compute. And it's also very plastic, means it adapts with experience. So every one neuron, as you go around and think and learn new things, and maybe remember a little piece of a conference or a lecture that you're at, your dendrites will change and your synapses will change. Okay, and that's how we remember the next day and the next year, and we form gradually our personality through our lifetime. Okay. So now that I explained how important these things are, I'm going to give you a very brief background about how we can go about experimentally to study these. And then basically this is the method section. This slide is the method section for the data that is deposited in neuromorpho.org. Okay, so then after this I'll stop with the slides and we'll start navigating the web together. Okay? So the idea is that first we have to visualize the neurons. There are now many different ways to visualize the neurons. You can find great reviews on, say, Nature Reviews Neuroscience or annual reviews of Neuroscience for a bunch of these techniques. One of the earliest techniques was bulk loading with silver, called the Golgi staining. The great Spanish neuroscientist Cajal used Golgi staining to illustrate or visualize a lot of neurons. So this is, in fact, neurons visualized with Golgi staining right here. But there are many other ways. You can inject dyes directly into the cell body and wait that it moves into the trees, both the input and the output. Now you can use immunohistochemistry, basically using antibodies to selectively attach, through the antibodies, stains and colors and labels to neurons. Or the most modern is genetic labeling, which is you can basically engineer fluorescence proteins in the genome of the animal, be it the mouse or the fly or the zebrafish or the worm, it doesn't matter. You can engineer fluorescence proteins, and now you don't even have to put a stain because these neurons are going to become green when we shine the right light on them. So we point a laser to the, either we make a little hole in the brain or we cut a slice, and then when we shine a laser, because there is a fluorescent protein, it's going to push light back out, right? And so we'll see those. So a bunch of different ways now to do this. And likewise, basically, Cajal had optical microscopes. Now we have great varieties of optical microscopes, including confocal, 2-photo microscopes that give fantastic resolution, signal-to-noise ratio. We have electron microscopes to do this, so there is a large variety of ways for visualization. And finally, once you see the neurons, you have to trace them to actually collect the data. And this is the kind of data I'm going to talk about for the next maybe 20 minutes, half hour. It's basically the structure of the neurons. You can think of it as coordinates, x, y, z coordinates of every little point in the branch of a tree. Right? So you can describe now the oak tree that you'll see in the park if you take a walk this afternoon, but now you can imagine the x, y, z position and the thickness of the branches of the entire trees. Okay? Okay, so let me jump into neuromorpho.org. So this is a completely free open website. We started this website 10 years ago. In fact, we just celebrated the 10th year anniversary. The version 7 that we released on September 1st, so about a month ago, is the 10th year anniversary. There are now more than 50,000 neurons in this website. Each neuron takes about a week of time, give or take, roughly, to trace. So when you actually count the number of hours that went into tracing these neurons, you'll see that I could not have done it, my lab could not have done it, all the people in this room could not have done it in a lifetime. So this effort, very much like GeneBank, is a community effort where we go around asking all labs that reconstruct neurons, please send us a copy of your data. We'll standardize, we'll put it here so that all data are in the same format. And now somebody like you can download it and start analyzing it and search through it and so forth. Okay? So you can read through it, and if you're interested in this, I encourage you to browse it. It's a fairly deep website. It has explanation and a little bit of the numbers. You can see the amount of data is growing quite dramatically over time. The number of categories or concepts to annotate the data also is growing quite dramatically, and the usage of these data also has been growing over the years. So this is the number of website hits that we're receiving and the number of downloads. Okay, so I'll go through some of these ideas. But there are many ways to get to the data, and all the data is open, so you can either visualize it directly on the website or download it on your computer and play with it. You can browse the data by animal species. As you see, there are many different species that are represented, from the worm, which is, the worm is the simplest species that has neurons, as far as we know, all the way to the human, and the human brain is the most complex brain that we know of. But the typical animal study subjects are rodents like mice and rats, and now the fly. People are very interested in studying the fly brain because it's smaller and you can genetically engineer it very easily. But as you see, if you're interested in knowing what the neurons of the whale or the giraffe or the salamander or the sheep look like, you're welcome to take a look. But you can basically browse by animal species, by brain region, by cell type, or by the laboratories that produce the data. Or if you're shy, you can just look at random neurons and let the archive pick some for you. Or you can look at the Neuron Atlas that I will illustrate in a minute. Okay, the Neuron Atlas works for the rodent specifically. Do you have any question here? Because I'm going to start browsing so that you see, but where I browse and where I click first or last doesn't really matter. So if you're curious about specific things, this is a good time. Or you can let me go for 5 minutes and then start raising your hands. Hey, try this, try that. I'm more than happy to improvise. Okay? So this is what the browse by animal species look like. I can click on any of these slices or any of these names here, and the numbers that you see are the number of neurons in each category. Or you can go down to this. I don't know if you want to look at chicken neurons. These are the chicken neurons. This is the list, in fact. If you now put your mouse on it, you see a preview of those morphologies there. And you can just, in this case, are, I don't know, probably about 30 or so chicken neurons. Okay? And you can do the same for basically any of the animals here. Okay? The browse by brain region and cell type basically look the same, and so does the browse by laboratory. As you see, there is one laboratory that is producing an enormous amount of data. That's the Chiang Lab in Taiwan. And what they found is a way to semi-automatically trace the neurons from the fly. Now, that's very interesting because a fly has a grand total of about 200,000 neurons. So when you look at the numbers there, we are over 20,000, which means they might be on their way to have traced already 10% of the fly brain. So it might not be too far before they do a sample that is equivalent to the size of the brain. Now, this does not mean that they trace all the neurons of one fly. This is done typically from many, many flies. Okay, so they probably have gone through about 20,000 flies to trace these 20,000 neurons. But still, statistically, it gives a nice picture. Okay? So if you click on any of these, so this is just a preview of the morphology, but if you click on any of this, you get to the neuron page, and the neuron page has a bunch of different aspects to it, so we're going to take a little time to go through it, so it's a long page. Basically has an image here, a virtual reality visualizer that allows you within your web browser to look at the neurons, in animation, and then the data themselves. Now, you can actually look at the data, and the data basically, it's a file like that, as I promised. There's a bunch of XYZ coordinates. So each row, it's the XYZ coordinate of a position in the branch. Now, as I said, neurons have pretty complex branch, so in this particular case, you have something like 8,000 tracing points or so. Okay? Which means that somebody under a microscope said click, 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 click 8,000 times. Okay? The structure of this file, you can look at the FAQ or the quick start, but basically is the number of the tracing point, the type of tracing point. So 2 is axon, 3 is dendrite, 1 is soma, and then there are a few more labels like that, so you can recognize the inputs from the output. And then you have X, Y, and Z in micrometers. And then you have the radius, so this gives you the thickness of the branch, and then the point that this one point is connected to. So you have, in fact, the whole topology of the tree all the way to the root, which is typically the soma. Okay? So you can do a lot of stuff, including a lot of interesting modeling. I'll show you some of the applications. Okay? Now, this is the standardized data, so it's the data that we ensure is in the right common format so that all the neurons are equivalently structured in the database. However, we also store the original data that the author sent us. Okay, so all those depend on how they reconstructed them, but we keep the provenance of exactly the original data in the experimental data. So if you see something strange in neurons, they say, wait a minute, what is this? You can go back to the original data always. Now you have to figure out their format, of course, but if you spend enough time, you should be able to, like we have, and then you can look at what the original data that came out of the experiment was like. And then you have the log files. The log files basically tell you everything that we changed from the original data to the standardized data, so that you see if we had to change something to, for example, center the neuron or reorient the neuron, you see those changes there. And finally, the log file, which basically says if there is something that is remaining in terms of strange features of this neuron, like long connections or very small radii that might or might not be real. Okay? So then we have metadata. Each neuron is annotated with the animal, the strain, if the original studies reported it, the age of the animal or the weight of the animal, the region of the brain that this was recorded from, the type of neuron, a lot of the experimental conditions, such as the staining method, the slicing direction, the thickness, and so forth, if there are any notes like this. And then the reference to the literature, so you can click on that and you basically go directly to the publisher site where you can download the paper if you have a license for that. Okay, where was I? Here. Now, we have a reciprocal agreement with the National Library of Medicine that publishes PubMed, so that if we have a link to their papers, and this is one of the most common search engines, they are going to have a link to ours, which means that from the PubMed itself, which is a very deep repository, you can automatically retrieve all the reconstructions from neuromorpho.org that are described in that paper. Now, this connection might be slow, but in this case, it's this one neuron, okay, that's described in that paper, okay? And then if you click on it, you go back to that original page. Okay, and then there are morphometric measurements that basically describe the geometry of the neuron with a variety of key descriptors. There is a lot more that you can extract yourself, and I'll show you tools if you're interested that allow to measure things from these trees. But these are the ones that we store in the database, so you can search through the database for neurons that meet some morphological criteria. Okay? So these are other examples of typical pages of other neurons. And now, if you click on the virtual reality animation, you first have to accept the security warning because it uses Java, so you have to say, yes, yes, I know that Java is dangerous, but then it will let you use it. This is software that we created, so I trust it, but I'll leave it up to you if you want to trust it. So this would be a typical reconstruction. So the green here is the dendrite, the gray is the axon. As I said, the axons are typically much bigger than the dendrites. And this was reconstructed from serial slices. In fact, if you put it perpendicular, you actually see the vertical lines. This corresponds to the experimental slicing procedure, so you can actually see them. Okay, and you can play with it, zoom in, zoom out, and you can also download it in some simulator formats. These are typical simulators that you can use in computational neuroscience to simulate the electrophysiology of the neuron, such as the propagation of the axon, action potential, or the integration of the synaptic input in the dendrite. Okay, so you can download it directly in those formats.
Could you show us a motor neuron?
A motor neuron, I would. You mean a cortical motor neuron projecting to the spinal cord, or a spinal motor neuron projecting to the muscles?
One that goes to the muscles.
To the muscles, I do, I think. Let's see.
Pretty long, right?
Yes, they are spectacularly long. In fact, let me think. We can probably search by length, and that might be the best way. That would be a good guess. Oops, this is keywords. Search by morphometry. Let's try it out. So we'll do it live if my speculation that they should be the longest. In fact, your speculation should be the longest. So let's say, we'll say length, and then let's see what the maximum length is. You can sample the length. So the maximum value here, okay, so let's say greater than 10,000. That's pretty mighty. So greater than 10,000 means that the total branching length is 10 meters, right? So let's say 5,000. Let's see how many we get. Oh, wow, a lot. Okay. 10,000. Oh, no. This is in micrometers. So if we want a meter, that would be 100,000, wouldn't it? Okay, that's more like it. 36. Okay, let's show summary. So that way you see, boom, those are motor neurons. So yes. Okay, so motor neurons. So these are actually dendrites. This is from the cat. I know this very well because I worked in that lab for a few years. But let's see, there should oops, there we go. So this is a pyramidal neuron. These are dendrites, these are axons. Another pyramidal neuron, motor neuron. Let's see, oh, there we go. So these are axons of motor neurons. So you see peripheral nervous system. So these actually go to somatic. So these are the axons that carry signal from the skin to the spinal cord. We're going to open a few of these. But let me see if I know that we have actual axons of motor neurons. No, those are hippocampal neurons. Okay, we should browse for a while, but for now, can we look at some of these projections to the skin? Is that?
Just one, fine.
Okay. So let's see. Do you want to open it up and see it in 3D?
Yeah.
Okay. So if you don't preload it, this is what you're going to get. Java, and then it says, yes, I accept the risk, and then you do run. And then you let it load the data. Hopefully, if I don't have too many of these open. I might have.
Is that a pretty large file?
Yeah, it might be too. I might have.
Carry on.
Yeah, that's quite fine. As I said, I'm more than happy to try it. Let me actually see if I do axons and motor neurons. So let me illustrate, because I have to illustrate all this stuff anyway. So let's try to search by metadata. So metadata is a very powerful search mode where you basically have drop-down menus for all of these species. So let's say that I want to do mouse, so I just clicked M. It goes down to all the animals starting with an M. By the way, if you see labels here, it means that we have data for those. We don't have labels for stuff that we don't have. And so let's say that we want mouse, and then we want as, let's say, brain region, we would want to have the spinal cord, I guess, if we want motor neuron. Oh no, this could be in the brainstem. Let me put the brainstem, because I know we have data motor neurons from the brainstem. So these are muscles that go to the ear of the animal. And now let's say cell type, principal cell, and then it will be motor neuron. Okay, so let's see if we have better luck here. So 85, let's do a organization by archive. So let me close a few tabs here so I find my way around. There we go. Okay, so these are in fact motor neurons. These are the ones I was looking for. These are from the Lichtman lab at Harvard. And let me see in the meantime. Okay, so the previous one that we had loaded finally loaded up. So yeah, no wonder. This is the axon that goes to the skin on the surface of the back of the mouse. So if you tap the mouse a little bit, it will. If you tap it lightly, it will like it. If you push harder, it will squint away. But these are the fibers that carry that signal. And when you actually look at the detail, all the way in, you realize why it took a little bit to load. These are probably 10 of thousands of points. So even if you look at the scale here, this is about the size of the back of the mouse. So maybe only a few millimeters, maybe half a centimeter. But the total cabling is many meters because of the branching complexity. Okay? And it's pretty flat because this is a projection to the skin. Right? So it's just on the surface of the skin. So it's about the thickness of the skin of the mouse. And so let's take a look at the Lichtman one. So these are just the terminal fibers that control the twitching of the ear. And they were traced using the technique that Jeff Lichtman invented called Brainbow. And it actually found that these fibers are very interesting because they send many parallel cables to the muscle. As you can see, this is, you'd imagine that the brain tries to minimize wiring, but at least in the motor neuron axon it doesn't. You see how there are many fibers that are parallel, and then they split this way. They go to the terminal, whereas they could just have sent one and splitting. So they actually have a nice paper discussing why that is. Okay. Please keep asking. Like I said, I have to illustrate stuff such as the metadata search or the morphometry search, etc. So these are great ways. For me, searching for retinal cells or motor neurons or hippocampal neurons doesn't make much of a difference. Any other question before we keep going? So I showed you the metadata search. I showed you the morphometry search. And I show you how to summarize the data by archive or by summary. At any one time, you can certainly download the data in bulk. So you can just say select all and then add to download cart or just get SWC files. SWC files are the morphologies. And then if you do that and you just get the SWC files, now it says watch out because the file size, the whole data you're downloading is 42 megabytes. Is that okay? And you say, sure, 42 megabytes is fine. Or you might say, if you try to download the whole database, you might run out of disk space. So you might be careful there. Okay? Okay. So the last browsing mode that I want to show you is the Neuron Atlas. So the Neuron Atlas right now only works for the rodent. So about a third of the data in the database is from mouse, rats, and so forth. And we collaborated with the Allen Institute for Brain Science that created a beautiful atlas of gene expression, but they also have a three-dimensional atlas of the brain. And we mapped the reconstructions of neuromorpho.org to the Allen Atlas brain so that now you can basically browse each of these little dots represent a neuron. If you put your cursor on, it will tell you will tell you up here which neuron it is and so forth. And you can left-click and open up the page of that neuron, or right-click and open up the neuron visualizer. And you can also use this window here to basically put in the actual anatomical surfaces of the neurons or remove the dots from those surfaces up to whatever you like. And this is basically the organization that we also follow for the brain regions, at least in the rodent in neuromorpho.org. And then it's color-coded, so pyramidal neurons are orange, interneurons are blue, and non-pyramidal principal cells are yellow. And you can basically, now I right-clicked. I actually right-clicked by my mistake, so it's opening up a neuron page. I don't know what I right-click. Okay. One random neuron, I guess. I didn't mean to do that. You can basically zoom in and out either by quickly clicking or using this bar, or you can rotate this, or if you ever get lost, oops, yes. If you ever get lost, you can use some of the saved views to go back to where a view that you're used to. And then at any one time, you can say, no, remove all the structures, they're too confusing, I just want to see the neurons in 3D. Okay? So this is a very nice way to to actually navigate through the data if you like, and it's been used in online courses, massively online, open courses, MOOCs, both in the Americas and abroad. So, and it's completely open. So, and I think I'm going to stop here with respect to the browsing of the data, unless you have questions. And I'm absolutely more than happy to show you more if you like. There are literally tens of thousands of neurons, so we could spend a week here and never be done. But very quickly, you can search the literature. So we have a literature database. And if you search the literature, you basically can put a PubMed ID, which is the identifier of let me close a few tabs here. It's the identifier of the article from the National Library of Medicine, or you can search from DOI, if you like, the digital object identifier for a paper. Basically, you can also view the publications by whether the data are available or not yet, which kind of reconstructions, or you can download the Excel file that has all of the bibliography. There are 20,000 or so entries in there, but you can easily open it with Excel. And now we also added a brief summary of examples of people using this data. So either using the data downloaded from neuromorpho.org, or papers describing the data that are deposited in neuromorpho.org, or of course references about neuromorpho.org, which are mostly from my own lab, and then just people citing data from neuromorpho.org. And this is a good place to go back to here. So this is just a summary of what we have in version 7.0. And typically, although laboratories all around the world reconstruct morphology to identify the neurons, once the morphology is deposited in a database like neuromorpho.org, you can reuse it for many different applications, no matter what the original goal that the authors had in mind in order to spend this week to reconstruct this neuron was. So for example, let's suppose that the authors wanted to reconstruct the neuron because they wanted to build a computational model of dendritic integration. So they needed the data for the neuron, they went to the lab, reconstructed the neuron, they published their paper on computational models because they wanted to test a hypothesis regarding synaptic integration in the apical dendrites, for example. Now, if they store the data, you could first reproduce their own analysis, which is always a nice thing in science. You could build a model, or maybe modify their model to test a different hypothesis, maybe studying synaptic integration in a different part of the dendrite, for example. Or you could use the same data for a different goal altogether. For example, you could mix this data with the axons from other data and study the connectivity between, or the potential connectivity between neurons of different types, between the axons and the dendrites. Or you could compare with geometrical analysis how different the dendrites from the pyramidal neurons in the visual cortex are from the pyramidal neurons in the motor cortex, for example, or in the auditory cortex. Or perhaps you're interested in looking at the difference between the neurons in the auditory cortex of normal subjects with the neurons in the auditory cortex of deaf subjects. Okay, maybe looking at the ones that are deaf from birth and the ones that became deaf later on. Or maybe an animal model of Alzheimer's disease or Parkinson's disease. And then you might have three groups. The original one, the one that has the genetic alteration that induces Alzheimer's, and maybe the one that has the drug treatment to try to alleviate the symptoms or cure that disease. Okay? And now you can see what is the change in morphology, for example. And many other such applications. So if you actually look and browse at the usage of these data in the literature, you'll see many. These are just examples. There are now nearly 1,000. We might have passed 1,000 now because this, I looked at this number, there were 900 early in the summer. So we might already have passed 900 articles based on data in neuromorpho.org. These are just examples. But besides the articles, they're also used outside of strictly academia, so to speak. So already 6 years ago, the Applied Math Olympia in China selected neuromorpho.org as one of the data sets for one of their hard classification problems. They wanted to see if their college students were able to do cluster analysis. And we got about 100,000 downloads in 3 days. There are a lot of Chinese colleges out there. Nearly crushed our server. But there are many other pointers to neuromorpho.org, including from Scientific Americans, from Neuroscience for Kids. There have been a conference on data sharing that highlighted neuromorpho.org. And there's been a testimony to the White House Bioethics Commission about data sharing focused on neuromorpho.org. Basically saying, it is possible to share data. Why are more people not sharing data? And this is just a flowchart of what we have to go through, database curators, to get the data into neuromorpho.org. We have to search the literature every day. In fact, we do it automatically, of course. But I receive emails about twice a week of saying, hey, there are these articles that contain reconstructions. I and my students have to annotate those articles, contact the author, say, hey, we see you have reconstructions. Can you send us a copy of the data? If they say yes, then we receive the data. We have to, with the authors, extract the metadata so that they're mapped to the metadata of the database so that you guys can search it and download the data that you're interested. And then there is a lot of quality control that goes in there. And finally, the release. We do about 2 releases a year. So this release was in September. The next release is going to be at the end of February. This is more like a developer perspective. OK, so I'm ready to switch gear here. Encourage you to read the terms of use. The database is completely open. You can do anything you want to do with this data except selling it, because the data is open and free. So if you download it and you redistribute it, you have to also do it for free. However, if you use it in publications, then you have to cite both the database, some of the papers describing the database, and the papers describing the data that you're using in your publications. Okay? All right, I'm going to switch gear and talk about Hippocampome. This is a much younger project. It was launched officially last year with a publication in eLife. And I'm going to spend a few minutes to introduce that concept. So the hippocampus is a part of the brain. This is a rat brain, pardon. The hippocampus is a part of the brain that underlies the consolidation and retrieval of autobiographic memories, of personal memories, episodic memories, as well as spatial navigation. So this morning I managed to come by myself from the hotel here, and I had a map, but I also had a mental model of where I was supposed to go, and my hippocampal neurons were probably firing like crazy. Now, the hippocampus has a very nice structure with layers and regions, and the way the neurons are described in the hippocampus is typically by saying where the dendrites are in those layers and regions and where the axons are. And I think I told you enough today to understand that where the dendrites are and when the axons are basically define the circle, right? Because the dendrites are the input, the axons are the output. So if I tell you where they are, you can figure out who can talk to whom. So you can basically classify neurons based on where dendrites are and where their axons are in given anatomical regions. And that will tell you what the potential connectivity of those neuron types is in the network. And this is a typical example of how neurons are summarized. This is taken from a paper from the entorhinal cortex, which is a part of the hippocampal formation that basically connects this portion here to the neocortex, to the entirety of the rest of the brain. Okay? So we created a knowledge base that basically takes this approach of saying all that we know about neurons in terms of where the dendrites and axons are, and simply catalog them, classify them, on the basis of their morphology. And this is called hippocampome.org, which is also a completely open website. The basic morphology matrix here is organized in the following way. Each row is a neuron type. So this is granule cell. These are various interneurons. This is the region of the brain of the hippocampus called the dentate gyrus, and it has 18 neuron types. There are several other regions. And there is a total of 122 neuron types that we have identified in this way. And then each column is a given layer of a given subregion. So these are 4 layers of the dentate gyrus. which correspond to 1, 2, 3, 4 right there. Okay? And if you see blue, it means that the dendrites of this neuron type are present in that layer. If you're colorblind, you should see a vertical stripe. If you see red, that means that the axons of those neurons are in that layer, or horizontal striping. And then if you see purple, or a cross, it means that both the dendrites and the axons of that neuron type are in that given layer. Now, this is a knowledge base, which means that for each of these neuron types, we have a page describing the entire neuron. I'm going to quickly show it to you, as well as evidence for each of these pieces of information that we're annotating that drills down to the original article in the literature and the quote in the article that shows you that feature. Okay, so we have 14,000 such pieces of knowledge. Sorry. So there are many ways to browse the data. One is by morphology, which is what you're seeing. One is by biomarker. One is by electrophysiology. So neurons are not just their trees. They have electric characteristics. So you can typically describe the electrical characteristics with a bunch of parameters such as the resting voltage, the input resistance, the time constants, etc. These are typical values for each neuron. If you put your mouse over, the cursor over, it will tell you how many data points you actually have. And if you click on it, you're actually going to get to that evidence. And the molecular one basically tells you which of the biomarkers, these are proteins or genes, like calbindin, parvalbumin, GABA receptors, enkephalin, etc., are expressed green or not expressed blue. Or if you see red or orange, it means that it might be expressed in the rat, not in the mouse, or expressed in the young animal, not in the older animal, or having the gene but not the protein and stuff like that. And it's all explained. The key thing is that from the morphology, you can get to the connectivity. So here is a table of connectivity. I should probably shrink it a little bit so you see the whole thing on the screen. And now the vertical list is the potential presynaptic neurons. The horizontal is potential postsynaptic neurons. And if you see white, it means that there is no overlap between axons and dendrites. But if you see black, it means that there is an overlap and the axon is excitatory. So that's a plus sign in the network. And if you see gray, that's inhibitory. And so that's a dampening signal in the network. And then green and red are the things that have been actually experimentally verified with electron microscopy. So there is a little Java for that, a Java applet that basically show you graphically all the various neuron types and their connections based on the scheme. And then if you click here, you get to the neuron page. You can also get to the neuron page by clicking anywhere on the browse matrices. And basically you have the name of the neurons, the synonyms, the articles describing those neurons, the morphology, an illustrative figure, which is going to take a little bit to load. Sorry, let me open it again. Okay. Then the biomarkers for those, electrophysiological data, basically anything you can browse, but now from the neuron point of view, and then the potential connections. And for any of these information, so for example, if we say that mossy cells in this particular case are, let's say, neurogranin negative, and I click on that, I get the evidence for that claim, right? In this particular case, this particular quote, in the rat dentate gyrus, mossy cells are immunoactive for calcitonin, gene-related peptide, but not neurogranin, for example. Okay? And basically you can do that for any of these data. You can do that for electrophysiology, and you might get the exact quote from the table, for example, table 1 of this paper. And likewise for biomarkers. If, I don't know, let's find something that is positive here. You can get all the quotes here for that. So basically you get the information about the characterizations of those neuron types, and you can build network models based on the connectivity. You have the electrophysiology that allows you to model the firing behavior of these neurons. And so you can model now from the cellular level all the way to the network level, at least for this particular structure, the hippocampus. Okay, as I said, this is a newer resource. It is also fairly deep, and you can find the references on the website. I'm also happy to live illustrate it for you if you have questions. But there is a lot more on the webpage of the center, which is krasnov.gmu.edu/cn3. And for example, if you click on data tools, you can download a bunch of different tools that would enable you to model and simulate some of these data. Any question? I'm going to close with just acknowledgements because I think it's essential. So both Hippocampome and neuromorpho.org are the effort of a very large team of people and very generous funding from research agencies. So in neuromorpho.org, we thank all the data contributors as well as the grant support. And so there are many labs that contributed data, obviously. And the people who did the work. So current members, many alumni of the lab that moved on to better things. And likewise, in the Hippocampome, there are many dedicated current members, but also many, many, many former members who moved on to different things. So I'm going to stop here because I think I have about 30 seconds left. And I'm going to have more questions, but I'm also going to be around. I'm more than happy to receive emails and interact by email. A lot of this stuff is obviously online and virtual. So pretty much anytime, if you like to do follow-ups, etc., we can also do screen sharing. And I'm thrilled to see more people using the data. There is a book published by MIT Press that came out last year. It contains a lot of this information as well as speculation and amazing pictures of both neurons and real trees. It's not too expensive, so I encourage you to read it.